Jahana. So the next speaker is Tobias Teichert, a monkey mesoscopic electrophysiology system to bridge the gap between cells, LFP, EEG, and BOLD. Tobias.
Thank you so much. And thanks to all of you for coming. I'm glad we could seat everybody. All right, so I have no disclosures. I'm going to start with stating that our brains are highly interconnected organs. Maybe not as much today after the big party yesterday, but at least in principle they are. And in order to really understand certain key properties of the system, we need to record from the entire brain at the same time, rather than parts of it in sequence. Now, that's easier said than done. And I think the first method that has really done that convincingly is fMRI. Right? The only problem is that it has a relatively slow temporal resolution. Now, if you look at EEG or MEG, they have fantastic temporal resolution, but given the inverse problem being as problematic as it is, the spatial resolution is relatively limited. So in my model system, which is the monkey, the rhesus monkey, we record single cells. And we've tried to address this problem by recording from as many single cells as we possibly can. And people have gotten really good at it. It's quite impressive. There's some labs out there that can record over 1,000 neurons simultaneously in one monkey while he's behaving. Very impressive. Until you kind of put it into perspective and notice, well, that's out of 135 million, right? So we still have a long ways to go, about 5 orders of magnitude. So what we try to do, essentially we ask the question, if we can trade single cell resolution for improved and bigger field of view. And so we did that by switching to a more mesoscopic neural signal, which is local field potentials. And so the idea was we wanted to build a system that is not targeting any particular brain region or any pair of brain regions. We wanted to target the entire, one entire hemisphere. And so essentially any brain region that you might be interested in, like cerebellum, prefrontal cortex, orbitofrontal cortex, hippocampus, we wanted to have an electrode within 2 to 3 millimeters of that brain region. And so here's how we approached that. You can see here the exploded view of the design. And I want to draw your attention to this function here. That's the grid, we're calling that. So it has 62 grid holes. And for each grid hole, we designed one guide pin and one 16-channel depth electrode. And after the depth electrode gets inserted into the brain, essentially it is designed so that the electrode spans the entire brain column underneath that grid hole, and the 16 contacts are spaced equidistantly between the dorsal aspect of the skull and the ventral aspect of the skull. So here's an actual view of the system, how it looks in real life. And you can see on the right hemisphere, over the right hemisphere, that grid with these 62 electrode locations. And this is after implanting the electrodes. You can see it gets a little messy. Fortunately for us, we just cover that whole thing in a cap, and then we don't have to open it for months at a time. And every time we want to record, the only thing we have to do is we plug a mini HDMI connector and hit the record button. So setup time is really negligible. So this is where the electrode locations actually ended up. So on the right bottom, you can see a top-down schematic of the monkey brain. The central sulcus is right around here. And each gray dot is the location of an EEG electrode. You can also see these horizontal lines, and those are the locations where we implanted rows of electrodes. And so for each horizontal line, you're then going to see a coronal slice over here. So for example, that's the most posterior one. And superposed over the gray matter outlines are the locations of the individual electrode contacts. Okay, so here's the system in action. So that's the response of the system in response to a white noise burst. And what you can see here on the bottom right is the EEG responses, starting with the P1 and then the N1. If you look at the intracranial contacts, you see auditory cortex light up. You also see beautiful responses in motor cortex, prefrontal cortex, and down here in the cerebellum. Okay, so now that the system is up and running, we wanted to use it as a bridge methodology, and I'm going to give you 2 examples, and the first example is going to be relevant for EEG source reconstruction. All right, so, and for that I'm going to focus on the frequency following response, also abbreviated as FFR. Now the FFR is an electrophysiological potential that entrains to the fundamental frequency of speech sounds. The example stimulus here is a Mandarin dipping tone. It sounds something like E. And below it in blue, you see the FFR response that we're recording from an electrode at CZ. Over the past 20 years, the FFR has been developed as a really important biomarker of plasticity and pathology in the auditory system. And so here's what we believe actually underlies the frequency following response. So every time there's a glottal pulse during speech, a pressure pulse hits your eardrums, and the auditory activity propagates through brainstem, midbrain cortex. And what we then record at the level of the EEG is the spatial summation of all these activities. All right, and so we're going to call this thing an FFR kernel, and you can think of it just like as an evoked potential in response to a single glottal pulse. Now, in real life, of course, you don't have single glottal pulses. You have whole speech sequences with many, many tens of these glottal pulses, and then the FFR arises as a superposition of these individual glottal pulses. Now, the tricky bit, and the reason why we're so interested in the FFR, is that it has, for one, it has very bad signal-to-noise ratio, and the other thing is that there's many, many simultaneously sources all across the ascending auditory pathway. And as a result, it has been really hard to source localize the FFR. And there's still very basic ongoing questions that are not answered. For example, does A1 contribute to the FFR? And if so, how much? And so that's the question that we figure we can answer really nicely with this system. Does primary auditory cortex contribute to the scalp-recorded FFR or not? What I'm showing you here is a map of the signal-to-noise ratio of the FFR. Two things I want you to take away from it. The signal-to-noise ratio is pretty high, right? So it tops out at about over 250. So we get beautiful signal-to-noise ratio. The second thing is we get these different spotlights, highlights of activity. For example, back here, we see an activity cluster in the brainstem. We see the inferior colliculus light up. Electrodes near the medial geniculate pick it up. And finally, we see auditory cortex. Great, so at least we know auditory cortex does respond to the FFR. We still don't know if it propagates all the way to the scalp. So what we did here is we plotted the FFR kernels for 5 different regions along the ascending auditory pathway. And you can see like different regions have very beautiful, very different types of responses. And if we plot the FFR kernel, our idea was it should be the weighted sum of all these contributions down here. And in fact, that's the case. So we can model the EEG kernel by a weighted sum of the kernels, of the intracranial kernels. And if you focus on that longer latency components, they really are nicely explained by activity in auditory cortex. Great, so we have these two signals that really match nicely, but we still don't know if that signal actually propagates all the way out to the skull, because it's a long way from auditory cortex to the skull. And to test that, we correlated the activity of the kernel in A1 with every single other electrode in the brain that we have. And we get these beautiful maps where you can see here that all the channels that sit above the superior temporal plane have a very strong positive correlation, and all the ones down below have a very strong negative correlation. In addition, what you see, as you go farther away from the superior temporal plane, the activity fades away with distance, but it still seems to propagate through the skull and is strong enough to activate our EEG electrodes out here. All right, so essentially what we're seeing here is a dipole in 3 dimensions. It's the first ground truth measurement of a 3-dimensional dipole that all of us model all the time if we do source reconstruction. Here's an actual one. It's the ground truth data. Okay, so the next example is going to be relevant, hopefully, for resting state functional connectivity. And one of the main questions that people have been asking about it is, what is the neural substrate, the electrophysiological substrate of the resting state bolt? And a number of different frequencies bands have been proposed to be relevant. And so the question that we can ask now is, well, does it even matter, right? So do different connectivity maps even depend on the frequency of the LFP signal, right? If it doesn't, then we don't have to worry about what's the actual underlying mechanism. Okay, so what we did here is we filtered our data into a number of different frequency bands, like a low band, the delta, alpha, beta, gamma, and so on. We also computed the envelopes. And in addition, we computed low-pass versions of the envelopes that match the frequency band that we would roughly expect from the bolt signal. All right, so we have these different ways of filtering the data, and now we're going to ask, does the resting state functional connectivity depend on the filter properties? And so what we did here is we picked a channel, a C channel in cerebellum down here, and then just show the C-based functional connectivity. As you can tell, you can see a beautiful connection between the cerebellum and motor cortex up here. All right. And let's see how different filter properties look like. So let's go for the beta low-pass envelope. And you can see quite a bit of similarity. You still see the motor cortex light up, but actually the pattern is much bigger. It's enlarged. It encroaches all the way up to the prefrontal cortex. And interestingly, you see these very strong negative correlations with the occipital and parietal cortex that wasn't there before. The next example I'm going to give is I'm going to switch the carrier frequency and now go to the delta band. And we're looking here at the delta low-pass envelope. And again, you see a very distinct pattern light up, right? You see now all of a sudden the cerebellum has a positive correlation with the occipital cortex and a negative correlation with parietal cortex. So from that, we would conclude that yes, the frequency band really matters and how you filter the data really matters. And it's going to be an open question how and which of those frequency bands contribute to the fMRI bolt signal. Okay, with that, I just want to conclude that I've shown you that mesoscopic electrophysiology, or MEPHIS, in the monkey is definitely possible. It's feasible. We get beautiful signals. And I think there's lots and lots of really interesting applications. For example, we can now go in and measure the ground truth data of these electrical fields that we like to model for our EEG source reconstructions. And in addition, we can now take a closer look and a deep dive onto the spectrotemporal properties of the resting state functional connectivity. More broadly speaking, I think MEPHIS is going to be a very cool Rosetta Stone methodology that can bridge the divide between a lot of things. For example, it can help us bridge activity between different scales, like from micro, meso to macro, different species, rodent, monkey, human, different recording modalities, such as electrophysiology, fMRI, all the way to optical, and different time scales. Now, and so with that, I want to thank my lab, and I want to thank you for your attention. Thanks.